In this episode of Voice of the Sea, we're exploring the Pacific Biosciences Research Center, PBRC, where students and their mentors are making biological discoveries, researching public health concerns, and protecting Hawaiian culture. PBRC supports Hawaiian and Pacific Island students across a diverse group of facilities within the School of Ocean and Earth Science and Technology at UH Mānoa. We visit some of PBRC's facilities to learn about research on copepods, mosquitoes, and mice, and how this work improves ocean health, fights disease, and protects native birds. First, we head to the Kewalo Marine Laboratory with researcher Kiana Frank to learn about the importance of microbes and how her lab studies them.
What I want to do in my research is try to understand our ecosystems better, just like our kupuna, so that we can get back to ecosystem sustainability and resilience. And I do that by looking at the tiny things that we can't see. A lot of my work is about uncovering the unseen, unveiling the invisible to our eyes, the akua of our Aiona, those tiny organisms that mediate all of these life cycles in the environment that help cycle nutrients, to help plants grow, or to help water be clean, to create abundance. For me, science is all about malama aina and how it's my tool for malama aina. How do I care about places, how I build relationships to places, and how I help to steward these places into the future. I felt like being faculty in PBRC, we think about ecosystems from the mountain to the sea, from molecules to ecosystems, to promote biodiversity, sustainability, and resilience. And basically, when you come down to the lab side, it really just looks like you're moving around small volumes of clear liquid, and yet it gives us a lot of information. And so in the background, you can see one of my grad students, manipulating small volumes of clear liquid to get at the who is there. If we're asking about the who of the microbes in Alo'i, we're taking lepo, we're taking mud samples. If we're asking who kane kavaiola, like the microbes in the water, we're taking water samples. So we go in helicopters all the way to the tops of the mountains. We have hung out of helicopters sampling waterfalls. And then, we go all the way down to the coast. And then we also go in submersibles down to the bottom of the ocean to collect samples.
Aloha mai kākou, aia mākou ma loko o ka moku lu'u o Alvin. Ho'olu'u.
And so my lab kind of runs the gamut across these ecosystems. We have the capacities to pull in seawater from the ocean, so we have fresh seawater, which allows us to rear live animals. And so we have coral rearing, we have squid rearing, when we have researchers here that look at biofouling and a bunch of different invertebrates, all the way down to my lab, which studies the tiniest organisms that live in the ocean, microorganisms.
A lot of people tend to get sick after swimming, especially like skin infections like Staphylococcus aureus infections. So that's been the major focus of my PhD and all my research. One major goal of mine, eventually with my career, hopefully even potentially with Kiana, is to focus on running an epidemiological study, taking water samples as people are swimming, and then follow the swimmers to see if they are getting staph infections, and try to deduct whether this concentration led to these many infections or increased the disease risk. Because there is no formal threshold, so we don't know what concentrations are safe or not to swim in. Right now, we're just making our best educated guesses and comparing one beach to another.
Next, we visit the Isabella Aiona Abbott Life Sciences Building to meet researcher Matt Medeiros and learn about the work his lab is doing with microbes and mosquitoes.
Microbiomes are these super fascinating and really, really complex microbial communities. There's hundreds of different microbes that can exist, for instance, in one mosquito, right? I've got quite a few students who are engaged in aspects of the microbial modulation of mosquito biology. Some of my students are working on microbes in Culex fasciatus and avian malaria. Other students are looking at different mosquito vectors that have impacts on public health, right? So dengue transmission or Zika or chikungunya, other arboviruses that are of public health concern. Our ultimate goal is to produce products that will be important tools in an integrated pest management strategy to suppress mosquitoes on the landscape. This has been done now in places like Fresno, California, in Florida, in Puerto Rico. There are examples of other groups using this technology successfully and safely in real-world landscapes to suppress mosquitoes that transmit human diseases. We're trying to do it to suppress mosquitoes here in Hawaii that transmit avian diseases and threaten the conservation of our Hawaiian honeycreepers. These birds, right? These are really important cultural pieces of our natural history. They are the kino lau of our familial deities, right? They are these natural resources that literally adorned our chiefly class, right? Their feathers were woven into the capes and the helmets that our chiefs wore. These are invaluable pieces of our culture. An act to save Hawaiian honeycreepers is an act to preserve native Hawaiian culture and the things that make Hawaii unique. So here's an incubator of our Wolbachia positive Culex fasciatus colony. So basically have a bunch of adults here. This particular colony was isolated here in Mānoa. So they proceed from egg to larvae to pupae to adult, much in the same way that, a caterpillar would. So this is the species that vectors avian malaria in Hawaiian forest. There is a large multi-agency plan to suppress this particular mosquito in areas where Hawaiian honeycreepers occur, largely because of their involvement in the transmission of avian malaria, which Hawaiian honeycreepers are highly, highly susceptible to. This is Aedes albopictus, probably our most widespread mosquito here in Hawaii that poses a real and acute public health threat or problem. Aedes albopictus, it was likely participating in the transmission of dengue in the 2015 outbreak. It was the sole vector for the 2001 outbreak of dengue here in Hawaii. It's known as the tiger mosquito. You'll recognize it. It's a very dark-bodied mosquito, but it has these very distinct white lines on its legs, and right down the middle of its thorax, it's sort of back. So this is a local Aedes albopictus strain that we're working with here. One of the really cool things about Aedes albopictus and other Aedes mosquitoes is that their eggs are actually desiccant resistant. They can stay dry and still be viable for several months to up to a year, maybe even longer. And this trait, right, this ability to have viable dry eggs has led this mosquito to basically colonize the world, right, through global trade and the movement of different things across continents. One bucket in your backyard, every week it can produce 1,000 to 10,000 mosquitoes if left unattended. But in terms of mosquitoes, no, man, I've never liked them. I still don't. That's the stuff of nightmares right there.
Right next to me are some bacterial cultures that I prepared this morning. And these are actually from bacterial stocks that I grew from mosquitoes that I collected over the summer. And so they were in this -80 freezer sort of in storage, and I put them in an LB broth to encourage them to grow. Eventually, I'm going to use these for a microscopy project. So we're going to use scanning electron microscopy to look at the morphology of these different bacteria that I'm interested in. And then I'm also going to use these bacteria for other experiments where I want to see how these bacteria influence the growth of mosquitoes and how they might be influencing different aspects of mosquito fitness. I'm hoping that the research that I'm doing can help support these landscape scale mosquito control projects, which are being used to help our native forest birds. Being Kanaka, who grew up in Oregon, I was always really interested in anything Hawaii, so to learn that we have really amazing special birds here that are really important in our culture made me really passionate about learning about them and then figuring out how we can help them survive. The other thing that really attracted me to PBRC and what I appreciate about the unit is that a lot of our research is very applied. So there's like a lot of focus around environmental issues, and that's really where I see my research going as well.
The University of Hawaii's Sea Grant College program focused on Hawaii's coasts and its communities through sustainable development, safe seafood supply, sustainable coastal tourism, hazard resilience, and healthy coastal ecosystems. Hawaii's Sea Grant.
You're watching Voice of the Sea. We're in the PBRC Békésy Neurobiology Laboratory with researcher Petra Lenz, learning about copepods, the tiny crustaceans that form the base of the aquatic food chain.
So the copepods we study are pelagic. So that means they're free living, they're in the plankton, and they're feeding on phytoplankton, the small plants in the ocean, and in turn they're fed by other organisms that are larger than they are. They're the base of the food chain, and just about every commercial fish that anybody is eating has either directly or indirectly has been dependent on copepods. The question that I'm interested in, how do they see the world? How do they feel the world? And how do they respond to it? And are they in a good environment or in the poor environment? When I started working on copepods, I was interested in the sensory systems and in their behavior. But more recently, I've returned to the field and I'm using molecular techniques to basically profile their physiological state. So what I'm using this new technology that has been developed called high throughput sequencing and of the messenger RNA, which gives me a relative gene expression, and then it can tell me whether the animal is in a good space, whether it's on track to complete its life cycle, or whether it is stressed in some way. So for example, if there are toxins in the water, then the gene expression will change in response. Or if there's not enough food, then they start changing how they express their digestive enzymes. So this is really a new research area. It's called transcriptomics. We are able to generate short sequence reads of all of the messenger RNA that's in the animal. Because remember, these are small, right? These are copepods. They're between less than 0.5 millimeter and 3, 4, 5 millimeters. And so they're easily ground up in their entirety, and I can get a profile of all of the genes that are expressed at a given time.
And I imagine like copepod populations, like they boom and bust over time, right?
Yes, and that's the problem. We know they boom and bust, but we don't know why. And that's been one of the problems with the more traditional methods of studying zooplankton, is that we can enumerate them, we can identify them. We can also use molecular tools to measure biodiversity of plankton. But trying to find out whether they are going to be there tomorrow or the day after tomorrow because it's a good environment for them or not is a lot harder. PBRC has been an incredible home for me because not only do we have the facilities here to do some of these studies in terms of the physiology, like the electrophysiology, and also the behavioral work, while being able to keep the cultures going, and also core facilities such as the electron microscope facility with amazing expertise. So we've been long-term collaborators with the staff at the electron microscopy facility in order to get this work done.
I've been working on drawing copepods, which is the main focus of this lab. This is one of the projects that I've been working on for the Heartline Lens Lab, which was a field guide here for students in Alaska. It will show all these copepods. So this is one of the copepods. It's a Microcitella. This is another important copepod called Neocalanus flamingeri. Another thing I worked on was a life cycle. I used Procreate. Normally I just looked at a picture. I try to make sure the proportions are correct. And then I try to make the colors similar to the image. And then I also try and look for like the little details. So I noticed there's like this little red part over here. So I tried to incorporate that over here. And I also noticed there's some red lines over here. And this is a lipid sac in the middle.
The whole thing is a lipid sac?
Mm-hmm.
Wow.
Yeah, it's pretty cool. What I like to shade my drawings to bring like dimension to the image. I want everything to blend smoothly, so I'm gonna just like overlap the colors with each other with a light hand.
How many copepods or different types of copepods are going to be in your final version of the guide?
There will be about 10 different species. There are a lot more copepods, but these are just 10 very important species.
We are looking for a few heroes, mentors, trailblazers, innovators. A passion to change lives, spark curiosity, open hearts, and awaken minds. Help students answer the question, who am I? This could be your calling, but this is no job. It's the journey of a lifetime. Be a hero. Be a teacher.
You're watching Voice of the Sea. We're in the PBRC Selenium Research Laboratory with researcher Lucia Seal. So tell me a little bit about selenium itself.
So selenium is a trace element. It's one of the periodic table elements. It's a micronutrient we need to eat a little bit in our diets in order to survive. It's essential for life. Usually green leaf vegetables and seafood are the primary sources of selenium in our diets. And so if you have a balanced diet, in general, you're going to have normal selenium levels. The selenium in our bodies that you acquire in your diet, it's then metabolized, and inside the cells, it's going to be used to produce what we call selenoproteins, which are the proteins that have selenium in its primary structure. And these selenoproteins are very few. We humans only have 25, but they are essential. And without them, we don't survive. And so that's why selenium is so important for our lives.
Is selenium something that is tested for in a normal suite of blood tests that you might get done annually from your doctor?
So selenium is not tested usually. Given the importance of selenium and the narrow range that we need to consume, that is a gap that we currently have in our health system. Also now you have first, supplementation. You go to the drugstore and you buy one kind of selenium and you use it, thinking that it's good for your health, right? Most people take to improve health. And the second part is that our entire food system, at least in the United States, it's a lot interconnected with fertilization, and fertilizers increase in general you are adding selenium to the soil or to the food where it ends up. So because of the way the food system is set up in the United States, we consider in general that the American population is mildly toxic in selenium, in its selenium status.
What happens if you have too much selenium in your diet?
Even if you are mildly toxic, your body has a ways to compensate and to excrete more of the selenium. Selenium is not going to cause a disease, for example, but it's preparing the environment. So a lot of the increased risks that we see, like in cancer, in obesity development, in cardiovascular diseases, that happens because there is an environment in the cell that favors the development of those diseases. The importance is really in understanding where the balance is being tipped, to what side this balance is being tipped. Is it favoring, in our case, favoring obesity development or preventing obesity development? And what are the mechanisms that are being disturbed when you don't have a proper selenium metabolism?
What I'm interested in studying is looking at how selenium deficiency and selenium metabolism affects cardiac function. This research is still in the basic stages. However, there's been reports of people that have heart failure that are associated with lower serum selenium levels and a poor prognosis. And so what we would like to do is, in the future, is to maybe tailor their diets for precision nutrition, to tailor their amounts of selenium in order to maybe compensate for their lower serum selenium levels. I love studying the heart, so I was studying it for my PhD. I did my PhD here at UH Mānoa.
I am an undergraduate research student here in the Selenium Lab. I originally joined this lab in October 2022 as a MARC program student. So MARC is the Maximizing Access to Research Careers program. I was very interested in biomedical research, and so the MARC program helped me connect with a mentor that I was interested in their project. And when I was looking, I was like, oh, I've never heard of selenium before. And I saw the impacts of what selenium can do, and I'm very interested in improving health surrounding diabetes and obesity. And selenium has various impacts towards those.
So tell me a little bit about your research in the lab.
My research project focuses on selenocysteine lyase, which is an important enzyme that helps recycle selenium in the body. When we remove that enzyme that helps recycle the selenium, they are having trouble and challenged in the cold room to produce heat and maintain their temperature, specifically for the males. However, for the females that don't have the cyclyase in the brown fat, they're able to produce and maintain their heat perfectly fine.
Oh, that's really interesting. Do you have a hypothesis why the females are still able to make heat?
I think that the males have most of their selenium in their testes, but the females have the selenium in various places. In the future, we would hope to use this experiment and hope this contributes to the health of humans.
PBRC is a center of excellence in interdisciplinary biological sciences with research, training, and outreach activities focused on the unique opportunities found in Hawaii and throughout the Pacific Islands. To learn more about the Pacific Biosciences Research Center, go to voiceofthesea.org. Follow us on social media at Voice of the Sea TV. Mahalo for watching Voice of the Sea. Exploring Our Fluid Earth is the dynamic curriculum developed by the University of Hawaii's Curriculum Research and Development Group. The award-winning Fluid Earth and Living Ocean textbooks are now interactive and online. New activities, updated content, and a teacher community. Exploring Our Fluid Earth is now freely available. Find out more at exploringourfluidearth.org.